So first off, before we talk about MPS and Ochratoxin, I really want to talk about chronic kidney disease, which is a global burden of disease. It's estimated 850 million people in the world suffer from some form of kidney disease. Risk factors for kidney disease include things like obesity, hypertension, type 2 diabetes, all of which are things which are increasing in frequency. To compare and contrast CKD and CKDu, so chronic kidney disease has a number of risk factors, like I just mentioned. What distinguishes CKDu from CKD, which is chronic kidney disease of unknown etiology, are things like living in developing tropical regions, exposure to high heat and humidity, agricultural environment, and probably the most important thing is onset in young ages, people that are 25 to 50 that do not have some of those other risk factors such as type 2 diabetes or hypertension. In addition to, in terms of how the disease presents itself, it presents with a lack of proteinuria and really appears to be affecting primarily the proximal tubules, where in chronic kidney disease, you're looking more at a glomerular disease from things like hypertension. So in terms of the global distribution of chronic kidney disease of unknown etiology, again, you see it primarily in developing parts of the world, such as Sri Lanka, India, West Africa. In addition, you may have heard of a thing called Mesoamerican nephropathy, which is another type of CKDu, which is you've got these hotspots of chronic kidney disease of unknown etiology. Again, these populations that don't have those classic risk factors for CKD. So what is Ochratoxin A? So OTA, it's produced by various different molds, such as aspergillus and penicillins. So it grows on food products that are stored under hot and humid conditions. It is nephrotoxic and it is hepatotoxic. It's classified by IARC as a class 2B carcinogen. Structurally, it's a phenylalanine with this isocumarine moiety. It's got a high log P. It's extensively bound to albumin, so very small free fraction. It's got a very long half-life in the body. There was a single PK study done in Germany with one volunteer that was given a tritiated dose of OTA, and the half-life was over a month long. Its major route of secretion is renal secretion, but there's actually very little renally secreted. And the reason being is OTA is a substrate for various different transporters that are involved in uptake back into the kidney proximal tubule, such as the organic anion transporters. In addition, because that high log P, it's also going to be passively reabsorbed throughout the nephron. And it's estimated from rat studies the concentration of OTA in the kidney can be 20 times that which you see circulating. So in terms of OTA food contamination, so it is ubiquitously found in food products. You can see wide ranges of OTA concentrations, up to this 20,000 microgram per kilogram of what you see here in this pumpkin seeds imported from China. It's interesting to note that only Europe and Canada have restrictions of OTA concentrations in food products, which are somewhere between 5 to 10 microgram per gram, okay? So in terms of human exposure to OTA, estimated concentrations of serum concentrations worldwide is about 0.7 microgram per liter, but you can see serum concentrations as high as about 2.3 or 2.7 microgram per liter, which is reported in a Tunisian end-stage renal disease patient. To date, OTA has only been associated with endemic nephropathy. It's not been directly associated. And the mechanism of OTA-induced nephrotoxicity remains undetermined. And this comes a lot in part from the various different epidemiological studies that are listed in this table here, looking at OTA concentrations in either people with CKD or normal healthy volunteers. And you can see in some cases in the healthy volunteers, the concentrations are lower, but in other cases, they can be significantly higher. And so there's going to be a lot of different factors going on, not only exposure, but probably genetics and other risk factors. So the important thing to know about CKDu is it a multifactorial risk type of disease. Again, exposure to hot, humid conditions, various different environmental contaminants, genetics, and also these farm workers take a lot of high doses of NSAIDs, which are also known to be nephrotoxic. Let's just point this out. Oh, back. So the biotransformation of OTA, this is probably very hard for you to read. Long story short, the biotransformation, oh good, I'm gonna be able to see my screen down here. The biotransformation of OTA is very complicated. And so what you see here in the center of this is the parent compound OTA, but it's really thought that the major metabolites of OTA have no toxicities. But having said that, there are some quinone intermediates which have not been identified in biological samples, but have been known to be generated in vitro. And these can redox cycle and form DNA and protein addicts. And so while most researchers think that the parent is the primary route of toxicity, it can't be excluded that metabolites also account for toxicity, especially in terms of oxidative stress and DNA addicts, because again, these quinones are very highly reactive intermediates. So I don't need to go into the details of the MPS system that we use in our lab here at the University of Washington. It's based on the Nordis microphysiological system. So I think they actually have a booth here at the MPS World Summit. So you can go to them and talk about the details of this system. But we've used this for a variety of different things. First, with NCAT's funding to develop a kidney on a chip to look at physiological function transport. And in my lab, we've pushed that for looking at toxicity testing. But again, we've also looked at various different functions, including in extreme environments like the International Space Station. So our working hypothesis is that OTQ, this quinone of OTA, which is produced by bioactivation of OTA, contributes to the nephrotoxicity. So this calls into the question the role of glutathione as transferase or GSTs in potentially mitigating OTA nephrotoxicity. Okay. So the way we actually approached this was just showing here in this scheme here, you're going to have, again, this is orally route of exposure from contaminated foodstuffs, uptake in the liver, first pass bioactivation by the various different cytochrome P450s. Then you can potentially get circulating OTQ or the quinone, hydroquinone, and they can be detoxified in the liver by GSTs, which can then actually be cleaved again in the kidney. Or you can even have direct bioactivation within the kidney itself by the resident P450s. And so the question we're asking is there a role for phase 2 metabolism in detoxification of OTA and the reactive intermediates? And so the approach we took here was to look at simply looking at chemical inhibitors as a way of understanding this in our MPS system. So in the case of the kidney, what we took about was we synthesized a pan-GST inhibitor called NBD-hex. And what NBD-hex does, depending on the GST isoform, has picomolar levels of IC50s. And so the idea was if we put in NBD-hex into a kidney MPS, inhibited the GSTs, that should increase the amount of DNA addicts oxidative stress and enhance nephrotoxicity. Conversely, if you put in ABT, 1-amino-benzotriazole, which is a pan-P450 inhibitor, if that P450 activation is necessary to get that reactive intermediate, that ABT should mitigate the nephrotoxicity induced by OTA. And so this is just a simple study designed looking at dosing MPS systems with various concentrations of OTA, ABT, and NBD-hex. And in this case, it's simple readouts looking at effluent biomarkers as well as a live dead imaging. So in this stain here, this is we've got four different stains here. We got bright field where you can see the kidney tubule. You got a live dead stain, which is a calcium AM for live, titanium bromide for dead, and total number of nuclei gets stained by DAPI. And what you can see in the presence of 10 micromolar OTA, which is a high concentration, but granted that level of that 2.7 microgram per mil in that one Tunisian patient is about 6 micromolar. So it's not out of the realm. But what you see here with this ABT and OTA, you don't really see any effects on viability. This is in contrast to that same concentration of OTA with NBD-hex. And maybe hard to see in the phase contrast image, the constituents of the tubule, but what you can see with the live and the nuclei is that really there's only little spots of color, because what happens in a microfluidic flow system, as the cells die, they slough off and are gone. And this is quantified here in this slide here. And so we can see in first with nothing exposed, we can normalize that to 100% viability. In the third column, looking at just treatment with OTA and ABT, you do see some protection going from 100% viability to about 70% viability. In addition, you see about 40% viability with OTA alone. But most importantly, in terms of the hypothesis of GSTs being important for detoxification, is that in the presence of 3 micromolar NBD-hex with 10 micromolar OTA, literally there is 0% viability. So showing that knocking out that pathway is going to be very deleterious for protection. In addition, you don't really see much compromise in viability with either NBD-hex alone or ABT alone. So when it comes to these MPS platforms, I always talk about these are not high content types of platforms, or these are not high throughput types of platforms. These are high content type of platforms. So one of our big readouts for high content is RNA-seq, so bulk RNA-seq on the chips. And so we looked at 2 different donors for different time points. So we looked at 3 days and 7 days, and we did increasing concentrations of OTA. And again, with these MPSs, we're seeding them with primary renal epithelial cells that we source from surgical waste. And so here's just a dose response looking at, on the left is going to be 3 days of exposure with an IC50 about 1.2. And with increasing exposure out to 7 days, that IC50 drops down even more significantly. And again, you can see the live dead stain with increasing concentrations from top to bottom, from 0 to 100 micromolar. You get very high toxicity at that 100 micromolar. And so here's just some volcano plots of the differentially expressed genes that we saw in this system, looking at either 72 hours on the left or 7 days on the right. And depending on 1 micromolar versus 10 micromolar, we saw anywhere from 2,000 to 1,000 differentially expressed genes. So to try to understand what's going on with all that huge number of data points, what we do is we do some pathway analysis using Advaita software. And of course, a number of different pathways come up, some which may not make sense whatsoever, like Parkinson's disease or things like that. But one of the things that did come up that was one of the top hits was glutathione metabolism. And so if you look at these different plots here, you can see, for example, a number of different GSTs are significantly downregulated in response to OTA exposure. Most importantly, one of the ones that's downregulated is GST-P1 or GST-Pi1. The reason why that's important is because that is the most abundant P450 in our primary renal epithelial cells, as opposed to like GST-alphas, which are very abundant in the liver. It's also important to point that out because GST-Pi1 is polymorphic. There's a common polymorphism in GST-P1, which reduces the activity by GST-P1 by 50% or even higher. And other pathways we looked at, we saw a number of different differentially regulated genes that were part of the NRF2 regulated cytoprotective genes. So we saw a number of these different genes being downregulated in response to OTA exposure. And so our proposed mechanism of OTA nephrotoxicity was, and we're looking at here, P450 bioactivation, whether that be in the liver or actually directly in the kidney, OTA to this hydroquinone, which then is very reactive, but GSTs can protect against that. And again, in response to OTA bioactivation exposure, you see downregulation of NRF2. And so NRF2 is a transcription factor which normally stays in the cytosol and bound with a protein called KEAP1. But under oxidative stress conditions, KEAP1 gets oxidized and dissociates, and NRF2 goes into the nucleus and regulates a number of different genes, including things like GSTs. So in summary, for this part of the work, this MBD hex's pan-GST inhibitor drastically enhances OTA-induced nephrotoxicity, while that pan-P450 inhibitor, ABT, had minimal or even rather protective effects against OTA toxicity. And again, under the conditions tested, neither one of these pan inhibitors had minimal effects. And again, this pathway analysis suggested that downregulation of NRF2 regulated genes by OTA treatment may help explain in part toxicity associated with OTA. So this led us to our next hypothesis, which is NRF2 activation will attenuate OTA-induced nephrotoxicity. And while MPS systems are our bread and butter, sometimes you don't need to use an MPS. One of the things we were talking about in the NCAT session today is use whatever is fit for purpose. So we thought, well, for this first screening process, let's just go ahead and use our same cell source, the primary renal epithelial cells. But in this case, let's go ahead and screen them in 2D first to see what's going on. And what we did is we used 2 different NRF2 activators to see if they could attenuate OTA-induced NRF2 inhibition. One is sulforaphane, which is a naturally occurring product found in very high concentrations in broccoli sprouts, for example, which is a NRF2 activator, versus TBHQ, or tert-butyl hydroquinone, which is a very potent NRF2 activator. But they both have the same mechanism where they promote the release of NRF2 from KEAP1. And so for this case, we looked at, set concentration of OTA, set concentrations of serofrane and TBHQ plus or minus OTA, 72 hours of exposure in 2D culture, and then did RNA-seq. So what did we see? So first off, again, what we see in response to OTA exposure, for example, glutathione reductase or GST-P1 are both significantly downregulated. We also see, for example, that a P1 is significantly upregulated by TBHQ. That's known to occur. But surprisingly, what we found is that in the presence of OTA, that these NRF2 activators, TBHQ or sulforaphane, that activity was completely mitigated by the presence of OTA. In addition, what's been shown when we look at these pathway analysis, OTA exposure induces a hypoxic response in our proximal tubular epithelial cells, or PTECs. And so if you look here in the bottom, in A, you're looking at HIF1 alpha. In response to OTA exposure, you see significant induction at the mRNA level of HIF1 alpha. Also, ARNT, which is aryl hydrocarbon receptor nuclear translocator, or also called HIF1B, which partners with HIF1 alpha, that is also significantly upregulated. In addition, we saw, I had this one enzyme here, this protein or enzyme here, BNIP3L was actually it's a receptor on the mitochondria, which is important for one of our hypotheses for OTA-induced nephrotoxicity. So this is one of the things when we do our tissue culture, standard tissue culture experiments, we're doing them wrong. Our standard culture conditions are 20% oxygen tension. Yet naturally, depending on the tissue, there's going to be much lower concentrations. So for example, here in the kidney is physiologically hypoxic. Renal cortex is about 30 millimeters of mercury. The renal medulla is one of the most hypoxic parts of our body with only about 3 to 10 millimeters of mercury. And so what we did is we cultured our cells under physiologically hypoxic conditions, in other words, normal conditions. Again, looking at this 2D culture and comparing the exposure of OTA plus or minus the NRF2 activators, sulforaphane or TBHQ. And here's something interesting to look at here. So under 20% oxygen conditions are standard conditions in the lab. You can see some translocation of HIF1 alpha into the nucleus as shown in green in the center panel with 10 micromolar OTA. Don't see much for ARNT. In contrast, when you look at 10% OTA plus 5%, so hypoxic conditions, you see a significant translocation of HIF1 alpha to the nucleus as well as ARNT. But in addition, what we also see, if you look at this under phase contrast, when you look at, for example, in the DAPI stain, the nuke blue, you see regions where there's no cells because it's actually quite toxic. So in conclusions, what we want to do is, we're defining the mechanism behind OTA nephrotoxicity because it's important for understanding the multifactorial causes of CKDu. It looks like our work indicates that nephrotoxicity may be caused by reactive oxygen species, OTHQ and OTQ. Current work indicates that NRF2 activation will not attenuate OTA toxicity. And finally, the message is, culturing your cells under low oxygen environment, in other words, these hypoxic conditions, plus OTA is going to be a more physiologically relevant toxicity testing. How are we doing on time? Okay, important thing here is acknowledgements. So first off, I'd like to acknowledge Jonathan Himmelfarb, who is the director of the recent director of Kidney Research Institute. He's a world-renowned nephrologist. He's been PI multiple of the grants involved in this. Again, I'd like to acknowledge the folks involved in doing the kidney MPS. Again, that's Moki Imoka, Jade Yang, and Nish Mahadio. Also at the bottom there, Kathy Young, who's got a talk later this week on using our MPS system for PBK modeling. The inhibitor studies were done by my colleagues in medicinal chemistry, Alan Reddy, and the bioinformatics was done by Theo Bamler, Lu Wang, and James McDonald, which are part of our NIHS EDGE core. And so again, acknowledging funding, NIHS, CASIS, and CATS.
Sorry, so the panel is open for questions, please.
I just have a quick question, Victor Hernandez from Lone Bio. In your model, when you were assessing the effect of the drug and with the live data assay, I saw a lot of the cells were round and you consider that as still live cells. Mm-hmm. If you change your analysis, would you have increased toxicity if you only consider the ones that are physiologically elongated and the right shape?
Right. I mean, so if I understand your question correctly, what's going on with the cells that are still alive? Yeah, I mean, so one of the other things we look at, I mean, I just showed you the live dead stain, but the other thing, the effluent that comes out of that chip, because it's a single pass, just like it is in your kidney. We also look at a variety of different biomarkers, urinary biomarkers that are associated with injury. And so we can measure those in the chips as well. And so if we look at early time points, we're not really getting acute toxicity by shedding of the cells. You can see release of those biomarkers, whether they be things like kidney injury molecule 1 or KIM1, or even urinary biomarkers, microRNAs we can see as well. So.
Any other question? With that, Professor Kidd, I have a question myself. So you mentioned about applying shear stress, about 1 din per centimeter square. To my knowledge, the proximal tubule, the apical side is exposed to 0.1 to 1 din per centimeter square. Why did you use 1? And then you're studying probably OAT1 and OAT3, which are basolateral transborders. So how the shear stress affects the basolateral area? That's my question.
Great question. So the 1 dyne per square centimeter is what I've seen is the average, depending on where you're in the proximal tubule. Yeah, and we're actually, I'd say we've got the S3 segment in that it's not convoluted. But I've seen numbers anywhere from 0.1 to up to 10 dynes per square centimeter, depending on blood pressure. So those are all variables. And so we picked, one because we're in that segmental. And in terms of the question about the basolateral transporters. So Great question. I mean, the way that we did these experiments, basolateral exposure is not the route of exposure, it was luminal. But there is a fair bit of OTA that winds up in urine and is actually filtered as well, especially when you have kidney damage. But we've also, we've done a number of studies using a vascularized system where it's very important to look at organic anion transport there as well. And we did some earlier studies using that system to look at, can we get transport of OTA from the basolateral side into the effluent. And that was a really easy experiment to do because OTA turns out to be very fluorescent. And so we just measure the fluorescence. So, but in terms of the basolateral transporters are absolutely critical in terms of understanding drug disposition. And so, for example, my colleague Kathy Young is me talking about the opioid PBPK modeling we did. That was, of course, all done using a vascularized system. And the organic anion transporters are absolutely critical for that disposition.
Thank you very much.